So as we are waiting, this webinar, I see some more people joining, so we'll give them a few more seconds, but this webinar is going to be recorded, and as usual, all of our webinars are available in our webinar series page, which I will show you in a second as well. So all the recordings will be made available there, and I think you will also get a link to the recording tomorrow automatically through GoToWebinar. During the webinar, if you have any questions, on your GoToWebinar panel, there should be a questions section, and feel free to type your questions in there, and we will answer them either live or just chatting back and answer, depending on the type of question. So, you know, definitely feel free to do that. Okay, I think we'll go ahead and get started. So today's webinar will be, again, as usual, our webinars, we try and mix between who's presenting. So today the presenters will include Evelina Bashenko, who's based at the University of Liverpool, Gloria Geraldo Calderon, who's based at the University of Notre Dame, myself, Omar Harp, based at the University of Pennsylvania, and Suzanne Warrenfels, who's based at the University of Georgia. And today we'll be focusing on VEuPathDB release 54, which just came out last week. And our goal with all of our webinars is not just simply to give you a rundown of a list of data sets, but rather we will demo some of them and highlight some of the cool tools and features that you can use within VEuPathDB to answer the questions that you may have using these new data sets. But of course, everything we show you today is applicable to any other data sets available within any of the VEuPathDB resources, since we're all built on the same infrastructure. So I'm going to go back here to the homepage of VEuPathDB that we're looking at, but the homepage of any of the component sites, so whether you're looking at Plasmodium or PyroPlasmodB or AmoebaDB, you'll notice they all look the same, and they have pretty much the same content. The only difference is obviously the data content, right? If there isn't a phenotype data set available for AmoebaDB, then you won't find phenotype data sets within this view of the data in VEuPathDB. I do want to highlight this section called the help section and the learn how to use VEuPathDB section. I think especially if you're very new to VEuPathDB, or actually for seasoned users of VEuPathDB, this is a place where you can learn about new features or learn how to use the resource most effectively. I want to highlight a couple of these, a few of these panels that you will find, hopefully you'll find useful. One of them is this FAQ, so frequently asked questions. So if you're ever using the resource and you're trying to do something and you're just not 100% sure how to do it, you might find an answer in our FAQ. So I would highly recommend that you come to this page, take a look at the questions, and if you expand one of these, you'll find more information about how to do it in a short vignette, hopefully that will guide you through running a step. So if you find yourself stuck, definitely come to the frequently asked questions and see if your question is available there. If you cannot find the answer to your question here, there are other ways for you to get help. One common way of people communicating with us is by contacting us. So you can click on this contact us link and then fill up this form. Basically, this will send an email to us, and we will answer you, and we do our best to answer you within 48 hours, so in 2 days. And questions range anything from how do I run an RNA sequence search in the database to I was examining results in a recent paper and I found that the expression values are different from what you have in a database. Can you explain this or that? You know, so there are many different types of questions. Also, we receive lots of suggestions from our community through this form, and I would highly recommend that if you have a suggestion for how to improve something on our site or a suggestion for a new data set on our site, definitely send us a note, and we love to hear from you. Another way that our community can interact with us is using this community chat button. If you look here at the bottom of the right-hand lower bottom of the screen, you'll see this community chat. If you open this up, this uses something called Gitter. You have to log in to start chatting, and you log in either with a GitHub account, which is easy to set up, or if you have Twitter, you can log in with your Twitter account. But you can see how all of these questions, these are available even if you don't log in. You can see different questions that the community asks. These are public questions, and the answers that are provided. So you can go in here and you can see a whole list of, for example, a threaded answer to a particular question. Those are a little bit more instantaneous, the answers. So we try, we get a ping whenever that happens. And I'm not going to say that you get an answer immediately, but within a few hours, you should be receiving an answer on Gitter. So that's another way for you to communicate with us if you want, if you prefer this way of communication. And then additionally, we're on social media, different places in social media. So if you're on Twitter, you feel free to tweet at us and ask us questions there, or on Facebook. We have a new component of our social media activities, and that is through Reddit. So I'll just go ahead and open this page here. If you have never used Reddit, I know Reddit has different connotations in different places, but it's certainly an open discussion forum, and these are places where anybody who's interested in bioinformatic resource center activities. So the bioinformatic resource centers include VEuPathDB, but also include the bacterial and viral bioinformatic resource center. So that's the second bioinformatic resource center funded by the National Institute of Allergies and Infectious Diseases. And so you can come here and you can find information about viruses. You can come and find information about Theileria, for example, what's new in the databases, and some other information here. Importantly, you can also post comments here as well. So that's another way for you to publicly tell us what you think or ask questions. And we are not necessarily the ones who may answer the questions. So the community, as we expand, the community will start answering these questions. So it's just another way for you to interact and communicate with us. Okay, so going back to the help pages here, I wanted to make sure to point out that, again, this webinar section here, I know I have the link. I had the link on the PowerPoint slide I showed, but of course nobody's going to remember or copy down that link. But the way you get to this is go under Help, learn how to use VEuPathDB, and then now you see this webinar button. When you click on this, this will open up all the webinars. So it includes our upcoming webinars, and this will continuously update this. So there'll be more updated in here over the next few weeks. But also, all of our previous webinars are available here. And so if you, for example, are interested in learning more about metabolic pathways in VEuPathDB, you can click on this link. This will take you to the section here about metabolic pathways, and you'll see here that there's a YouTube link to this webinar. You can access our YouTube channel as well from up here. So in addition to all the webinars that we've been recording over the last couple of years, we have specific tutorials that are there as well that you may be interested in exploring. Then the last thing I wanted to show you before I turn it over to Gloria to talk about some cool new vector-based data sets, I wanted to point out to a new thing we added in this release, and that is a running list of data sets that we are working on. So you can always go and go under data and look at data sets in VEuPathDB and explore all the data that's available in VEuPathDB. This is available in VEuPathDB to see everything in the whole world of VEuPathDB organisms. Or if you're only interested in Plasmodium, you can go under this filtered view, basically, of VEuPathDB, which is Plasmodium, and you can go and look at the data sets that are in Plasmodb, for example. It takes sometimes a few seconds to load, but and you can filter on this, and you can, for example, say Rna-Seq, or you can filter on a particular person, a particular user. So, if I knew there was a data set by Reed et al., and I'm trying to see which one it is, and then you can see here, oh, these two data sets are here, they're available or not available. The other thing you may be interested in knowing is, well, what data sets are we scheduling in our pipeline? And for that, if you go under Submit Data, we have some information here about how to submit data and what our data release policies are. But also, we have this data sets we are working on. And so if you open this, this will, again, load an embedded table in here. And this table gets updated. We'll try and update it frequently because we're continuously scheduling data sets. And you'll notice here that this was updated on September 7th. And then here's a running list of data sets that are on our horizon and the projected release. We release once every 2 months. So the next release will be sometime in November. And so you can kind of get an idea of when these are coming out. It's not a guarantee, obviously. Some data sets will have to move because of either issues we discover in our pipeline, or maybe some data is missing, or maybe it's not ready for release. And so, but this is a nice way for you to go in and quickly see, well, I know that there's a Canada data set that just came out on whatever, Canada population biology. I wonder if it's there. And so you can look for it in this table and at least see it. Again, this is a nice way for you to know that, oh, well, they don't have listed this very important data set for the Cryptococcus community or for the Toxoplasma community. I'm going to let them know. And so for that, definitely contact us and let us know about the data set. This will help us tremendously to make sure that we are on top of what you think are very important data sets to be included into the resource. Okay, so with that, I'm going to switch over to Gloria. So Gloria, I will make you the presenter.
Thank you, Omar.
Go ahead, you're the presenter now.
Thank you very much. Hello, everybody. So as Omar said, I'm going to show you what is the new data that we have in Vectorbase for this release. So here, if you go to the home page, you can notice release 54 from the 8th of September, and you come to this side panel here. Here you click news and tweets and release 54. What is the new data that we have for Vectorbase? We have 2 new genomes, Anopheles colutzii mopti and Anopheles arabiensis dongola 2021. Both of them for now without annotation, but it's already in our pipeline to have annotation for our next release. So I will save the demonstrations about these genomes for our next webinar. Today, I am going to focus on the population biology data. For this release, we have 7 new projects, and for 3 projects, we have some minor fixes to do. How can you explore and use the Vectorbase population data? So you can come to Tools and click here Map View, which is the tool that we have developed specifically for you to look at the data. Because we are in the release notes, you can click on View on Map. So for each one of these specific projects, you can see how they look. And notice that each one of them has an ID, Vectorbase project, and a consecutive number. So if I load each one of these codes for each one of these 7 projects, and I type them all here in the map, so it's telling me for all the 7 new projects that we have in Vectorbase, these correspond to which available data types: to abundance, to insecticide resistance phenotype, and to insecticide resistance genotype. And the little icon right here is telling me I can switch the view and not just have all the samples available, but to one specific that I want to look at. If I, for example, I can unclick any of these samples or just delete them all, and this table right here is just an overview of everything that we have in Vectorbase, all the available data types. Okay, going back to the 7 projects that we have for this release. So this one, I'm going to start with the abundance view. So I'm looking at here with filters that I can use on here, this right-hand side panel, or here on the left. So I can select a point, for example, this one, and notice how I get this yellow halo here. And I'm looking at the species that correspond to this sample in the abundance. I can also look at the specific metadata for this collection, when was collected, how was the collection protocol, how was the identification protocol. And I can also use that information and say, okay, I want to look more into that, into the protocol, for example, collection protocol, that's what I was looking for. And it's loading right there. And notice Now I have gray and no colors. So I click on optimize colors, and I can see for this point that I selected all the different protocols that we use to catch mosquitoes, or any vector that you're using here. So this is for abundance. For this release, also for the 7 projects, we have insecticide resistance phenotypes. So again, I can select a point in the map, and right now it's telling me which species I have available data. But I want to know which insecticides were tested. So I change right there, and it's telling me all these different insecticides were tested. And I can see, with this one, what is the level of resistance or susceptibility that I have. For example, mortality rate, 95%. And here on the other side is showing me only 13%. So what we are looking at here is this, the data of all these 7 projects. The background is all these 7 projects against only the sample that I have selected. And the last one, for this release, we also have insecticide resistance genotypes. So again, these are the species that are selected, and I want to know which alleles we have data for. So it's showing me both the reference allele, for example here, KDR, and also the mutant allele, because notice the reference only has the amino acid at the beginning, but the mutant has the amino acid at the beginning and at the end. So you can check on that. Something else that I want to show you today is that you can download the raw data. So you do not need to communicate with the data provider, because they already deposit the data in Vectorbase. So you can come here and say, well, for these 7 projects, or for whatever you are looking at the map, which data, which metadata you want, so you can work with the raw data. And here I have some 3 examples of what people do with our map. So for example, they said they collect microsatellite and Snip genotypes, and they deposit the data in Vectorbase. And in their paper, they provide the project. So you can type those codes and use them in the map. And here, for example, they said all the microsatellite database of Aedes aegypti, and how many genotypes and how many populations corresponding to which countries, and that everything has been deposited in Vectorbase. So it's a nice view to tell your readers how to interact with your data. And this, for example, was a study of, population study of Aedes aegypti. Some other users have the data from the map, and they actually do what I just told you. They download the data to create meta-analysis. So for example, in this case, they are using 165 studies with terrestrial and freshwater fauna. And, they are just, using one, some of the studies that they analyze here are coming from Vectorbase. And also they generate review papers. For example, here, data on disease vector interactions were extracted from Vectorbase. So they're just using the vectors, Aedes, Anopheles, Culex, Culicoides, Glossina, Phlebotomus, Simulium, and they are looking at host biting patterns. And this paper is a network perspective on human diseases. And they are using what I didn't demo today because it's not in the release, but is the blood meal source. So these are the different data sets that we have new for Vectorbase and how people use them when they interact with the map. That's all I have to say.
Great, thank you. So we'll switch over now to Eve, who will tell us about updates in FungiDB. Eve, I'll go ahead and make you the presenter. Oops. Okay, you should be the presenter now.
All right, thank you, Amar. Let me just, yep, you should be seeing my screen now.
Yes.
Okay, so I am on the main landing page for FungiDB, and as Gloria showed you, you can easily access the news releases right in the right panel. And in this release, there are several new datasets for fungi. For example, we have updated the genome of Trichoderma rhizii with annotation, and the authors used PacBio and Illumina platforms to create high-quality genome assembly. Now that the gene annotation has been loaded, you can mine this genome with gene-specific queries, and if you're curious in terms of what type of queries are available, you can navigate to the dataset record page, just like I'm showing now. Scroll down to the bottom of the page, and the available list of searches will be identified for you within the Explore this dataset within FindGTB section. Now, coming back to the news, we also have several new Snip datasets, and these datasets are pertaining animal and human pathogens, such as blastomyces precursors and also Cryptococcus neoformans. For example, for blastomyces, we know that for quite some time, blastomyces genus was thought to be comprised of a single species, Blastomyces dermatitidis. But in recently, 3 other species have been described, including blastomyces precursors. And in this release, we integrated whole genome sequencing of clinical data of blastomyces precursors, which causes blastomycosis disease in South Africa and Middle East. We have also added a new sample to the existing data set in Cryptococcus neoformans because it became available in this array, and now you can browse the full spectrum of this data by navigating and creating different type of searches that utilizing Snip data. And similarly, you can identify the type of the queries available from the dataset record page. Now, in addition, we have one more data set that I wanted to mention for Cryptococcus neoformans, and this is a data set where SNPs were called on WGS of clinical isolates related to cryptococcosis outbreak in Glasgow in 2018. And similarly, you can access this data set either from navigating to the dataset record page, or you can deploy a Snip search by essentially start typing SNPs and navigating to the different types of the queries that use this data set. Now, when it comes to the transcriptomics, and I'll scroll down a little bit further. When it comes to transcriptomics, we have several data sets for you now, and these data sets were loaded for Coccidioides, Fusarium, Mucor, and Neurospora. All of these data sets can be accessed under the Rna-Seq Evidence Search. And for example, if you would like to find these data sets, you can simply start searching RNA, and the Rna-Seq Evidence Search will come up within the results. And if you click on the Rna-Seq Evidence, you will be redirected to a list of all data sets that are currently available in FindGTB, and all new data sets will be also clearly labeled, such as this one for Coccidioides immitis, where a range of searches and the type of the searches available is indicated by the buttons here. For example, this is a differential expression search, fold change, percentile. For example, you can identify genes in the 90th to 100th percentile of expression, and sense or antisense strand RNA. For those data sets where replicate data is not available, you will notice that the differential expression button is missing, is not available. And obviously for those data sets that are not stranded data sets, the sense antisense option will not be as well available as well. But for this one, if you would like, for example, to utilize the sense antisense search, and that could be simply in the contents that we know that the natural antisense transcripts and long non-coding RNAs have been reported in fungi before. And for example, if you would like to identify genes that may be regulated by antisense RNAs, you can simply create a search when the search will be constructed, such as you want to identify genes whose antisense transcript are increased while the same gene's sense transcript are decreased. And this particular sample is looking at different developmental stages of coccidioides, such as mycelia, mature spherules, and young spherules, and the difference between these two, that mycelia is a non-pathogenic sort of form of existence for coccidioides, while spherules is the pathogenic form and the disease-causing life stage of coccidioides. And so you may want to choose any and all of these samples to identify all possible combinations. Or if you want to say, I want to compare only two, compare the mycelia, which will be my non-pathogenic or control sample, versus mature spherules or pathogenic stage, and just click Get Answer. The search will essentially look for anything that fits your criteria and return a list of genes. And so if you examine the list of genes, you now can see that the search identified 148 genes, and those genes are listed on the table within the table below. And as you scroll down to the right, you can also view the genes and their graphical representation, such as TPM for both strands for this data set as a graphical representation within one column, and the column right here on the right actually summarizes which samples fit your criteria, such as the search that identified, for example, samples that fit the mycelia versus spherules come up and are designated by the sort of red square right here at the top. And so this is the sample that came up in our search, where we know that there is immature spherules. This particular gene has an increased expression of the antisense transcript versus the sense. And so coming back to the news back again. As I have mentioned, we have several RNA-seq transcripts, RNA-seq data sets where the transcript can be searched using the various RNA-seq evidence tools, tools that can help you mine this data. One more thing that I did want to mention is the update on the manual curated data sets. And before, VEuPathDB has this data as separated in the project. We curate several data sets in-house, and previously all manually created phenotypes were grouped based on the species. For example, we had a group for Aspergillus, Cryptococcus, Fusarium, and Parvicularium. In this release, all of these records were merged into a single project that is called Manually Created Phenotypes. And from this point on, any records that are updated within the currently curated genomes and also future curated genomes will be integrated here. And that is also simplifies how you can search and mine this data within FungiDB. And I believe this is all from me at the moment, so I will pass it back on to Amar.
All right, thank you, Eve. So next up will be Suzanne, and she will cover actually a number of databases. So I'll turn it straight to her to describe those data sets. Suzanne, it's yours.
Okay, thanks. Can you see TriTrip on my?
Yes.
Am I sharing my screen? Okay, cool. All right, so yeah, thanks. As Omar says, I will give an overview of the new data in TriTripDB, ToxoDB, Microsporidia, and Trick. Hopefully I can do that in 15 minutes. Right, so here we are at TriTripDB, and I'm going to go to the news where all of the new items for that database are listed. And under new data, I see that we have 2 new Leishmania genomes that are annotated genomes. And we'll come back to those, and I'll demo a little bit about the HU3. In this release, we've also updated some functional annotation. And the first thing that we wanted you to know is that GeneDB has been taken offline effective August 1st. So don't worry if you were someone who used GeneDB, because everything is still available in TriTripDB. And we are still using their Sanger FTP site. So you can visit that site and download large-scale files from them, as well as our download data files section also. So our curators have updated 4 different genomes with new functional annotations, and here's a table that gives a summary. We also have a Google Doc for delving deeper and learning more about what those functional annotations were. On this first page here, we just give an overview of what fields per organism were updated, and then within each of these other pages, you can see exactly what was updated for each of these genomes. So if you're an annotation person, you might want to look at that. Our curators also look at your user comments, and any comment that you enter on a gene page is looked at by our curators, and when it's appropriate, they integrate that into the official annotation for that genome, and that occurred for 11 different genes across 2 organisms. Okay, so let's look a little bit deeper at L. donovani HU3. That, of course, all of our data sets get a data set page. I linked there from the news, but I can also use the data sets in TriTripDB that Omar showed you earlier in Plasmo. And then this, is an overview. It has the paper, description, all the folks involved, links to every search available on our site, more applicable links, and then statistics about the annotation at the bottom. And so when I looked at this data, I was interested in HU3, what's new about it. I learned that it was sequenced with a combination of Illumina and PacBio methods, which is great because now we have long read sequencing, which helps give a better assembly. And these guys really wanted a great assembly, so they did it 3 times and produced a 36 chromosome assembly with no gaps, 0 gaps. And the annotation was performed with Companion. They used L. Major Friedland as the reference. Okay, so let's look a little bit. Well, so first I want to show you a search based on the annotation, and then we'll look at a way to take advantage of the orthology profiles to learn more a little bit about Leishmania-Donovani HU3, even though we don't have expression data in this in TriTripDB yet. So first, let's look at a search based on annotation, and I will go to genes, function prediction, and go terms. So gene ontology is an annotation tool for assigning functions to genes. The ontologies are controlled vocabularies that can be applied across all organisms across the tree of life. And since HU3 is annotated, then we know at least some of the Go terms applied to genes. And then we at VEuPathDB built a search for investigating those annotations. And that search is right here. I will look for, I want to search HU3 for, let's say we're interested in kinase genes, genes with kinase activity. I'll choose protein kinase activity. You notice, whoops, you notice that there's many different Go terms that include the word kinase. I'm just going to choose one and search that. But you can, if you're really interested, go back and search all of them at once or just a few. And what I find is there's 211 genes that were tagged with the Go term protein kinase activity, right? So that's interesting to know. And if I want to delve deeper, I can go to one of these genes and go to the Go term table here. And I find that not only protein kinase activity is tagged for this gene, but there's other activities associated with this gene as well. And I can delve deeper into those, look at other things on the gene page, perhaps protein features and properties might have some information about this gene. So I find that there's other protein interpro domains that are assigned to that, which might have a clue for activity. And I see that there are no transmembrane domains or signal peptides associated with that. So that's a way to search the annotation and then to go further into looking at genes associated with that annotation. So let's go ahead and look at the new Rna-Seq data. I'll go to the Rna-Seq search page and filter by Donovani, and I find that there are no Rna-Seq data sets available that are aligned to Donovani HU3, but there are ones aligned to Donovani BPK. So let's go ahead and search that, and then we can take advantage of the orthology profiles to look for genes in HU3. So this experiment compared visceral to cutaneous transcriptomes, and I'm going to look for genes that are upregulated at least twofold using cutaneous leishmaniasis as the background. I will look for upregulated genes in visceral leishmaniasis patients. And what I find is 2,000 different genes that are upregulated. There is another search available for this gene, for this. All right, okay, I'm sorry. So what we find is 2,096 genes that are upregulated. You know, we could go back and edit this and find, you know, larger fold difference genes if we wanted to reduce that number of genes. But the problem that we're experiencing is that all of these genes are Donovani BPK genes, and we're interested in actually looking at HU3. Well, when we integrated this genome, we determined orthology profiles to every organism that we have in our database, and so we can transform these 2,000 genes into their HU3 orthologs. And you do that by adding step and then choosing transform into orthologs. I'll choose HU3 and click run step. And what I find is that there are 2,383 HU genes that are likely upregulated in visceral leishmaniasis versus cutaneous leishmaniasis. So that's a way to infer data for new organisms that don't yet have RNA-seq data. Okay. So I'd like to move on to Toxo. And I'll go to the news section there, just because we have an overview of everything that's been done. I find that we have, for new data in ToxoDB, we have a new version of the N-Canonum Liverpool genome that has been sequenced again with PacBio. So it's you know, it's a very good assembly. And we have a transcriptomics data set that is stage-specific RNA sequencing in T. gondii, and that's part of a larger study that was published in Cell in 2020. So let's go ahead and look at that data. We can search based on genes for RNA sequence data, and the new data sets are pretty easy to find. They have this big one here, icon here that says new. And our differential expression search applies statistics to the data. And since statistics are always a wonderful thing to have, let's search based on that. And we will look for genes that are upregulated at 48 hours in bradyzoites compared to tachyzoites. We want upregulated with a p-value of at least 0.001. I can say get answer. And I find I have 1,281 genes, and that's a lot. So I want to start looking at the data that we've accumulated for these genes and are showing you in our results table. And the first thing I notice is that some of these genes have fantastic p-values, right? And the second thing I notice is that this one here is hypothetical, right? It does not have a product description. So there's an opportunity to learn something about this gene. So I am going to go to the gene page and then to the transcriptomics section. And the transcriptomics section opens with a summary table, which is just a great way to get an overview of what your gene is doing in these different experiments. And our experiment is right here. I can click this to turn on the labels. So in tachyzoites, the expression is very low, but in bradyzoites, the expression is very high. And then just by eyeballing this, we can say, wow, I mean, there's a big difference here, whereas in these other data sets, not so much. So if I want to look at this more, I can go down to here and look at the full description of this data set, where there's a data table to look at these numbers. I can turn on the percentile graph to look at it in a little bit different way. And then because I'm on the gene page and I'm interested in this gene that maybe we don't know a lot about, there might be other clues elsewhere in this on this gene page. So I'm going to go ahead and go to the function prediction section, and I see that there is a Go term assigned to this gene. It is, and it's an integral component of the membrane, which is interesting. I can also go to the protein features and properties section, and I see that it has a transmembrane domain. So those are clues to what might be an interesting gene for someone to study.
Suzanne, out of curiosity, I wonder if you scroll down a little bit and look at the protein load targeting, did it have any data over there from the hyperlipid experiment? Let's see. It looks like it does not. That was, I was curious if it actually had any localization, but it doesn't. That would have been a nice feature. So
yeah, that'd be super great, right? And you might actually also see that in the Go terms too,
right?
With the cellular component. Yeah, that would have been great. Okay. Okay, so to recap then, we searched this new data set for statistically significant differentially expressed genes. We found that one that was differentially expressed with a high p-value, but relatively unknown. And then we went to the gene page to look for clues about what this gene might be doing. So that's what I want to show there. And then I will just mention that in VEuPathDB, we integrated a new encephalodazune Helm Swiss genome. It's the same sequencing, but it's been reassembled with modern methods and re-annotated. So it's quite an improvement over the old one. So if you're interested, you could check that out. And then in TrickDB, we have also integrated a genome sequence of a second organism, Trichomonas tenax. So which, because it's not annotated, many of these searches are not available, but you can still blast against that if you like. You can blast against the genome to find if you're, where your gene would be, and then you can look at it that way. So that's all I had.
Great, thank you, Suzanne. I'm gonna
already.
Then. Let's see, there we go. I'll show my screen. Okay, so to wrap things up, there are a couple of more data sets we'd like to highlight in some of our databases. So Suzanne already told us all about genomes and how you can take a look at genomes. And I just wanted to mention that in AmoebaDB, we loaded the genome for Mastigamoeba balamuthi. Of course, you may be thinking, well, why should I care about this organism? But this is an amitochondrial organism, so no mitochondria. So it's doing some cool biochemistry in its cytoplasm that typically takes place in the mitochondria. And so from an evolutionary perspective, it's quite interesting. And you can obviously, it's also related to Entamoeba. So that's a connection there where you can then use it as a model organism for understanding more about how mitochondrial biochemical activities are taking place in the cytoplasm or other places in the organism. This is a fully annotated genome coming from Jan Tekesi's group. And again, all the searches, I won't highlight them again, but Suzanne mentioned many of them, but you can look for genes based on all the features that are available, both from the annotation that came from the Tekesi group, or through the analysis pipeline that we run, which basically does things like looking for genes with signal peptides or transmembrane domains or assigns interpret domains and so forth. So that becomes a very powerful tool. I'm going to jump to Plasmodium and highlight a couple of new data sets. Again, in the news section, this is where you can actually quickly see what is new. So we've loaded a few new data sets. One is a Plasmodium Bergeye RNA sequence data set comparing parasites that were infected into mice that were experiencing caloric restriction or not. And so, as we know, there is a potential connection between diet and transmissibility and growth of the parasite. The other one experiment, and this is coming from Maria Mota's group in Portugal. The other data set is Plasmodium vivax data set that looked at samples, patient samples grown in culture for a few hours right after thawing them from the freezer in different medias. And so if you can always look at the paper and see how they organized the experiment, but you can see here they basically took these parasites from the freezer, they thawed them in different medias, and then collected time points at different points and looked at their expression panels, and these came from 3 different isolates. And again, I won't go into the details of this, but you can search it just using similar searches that Suzanne did. And the third data set is a proteomics data set, and this is a quantitative proteomics data set that compared Plasmodium falciparum parasites with a shield knockdown of this CRK4 gene. And so they compared the wild type to the shield treated. So plus shield minus shield. In this case, it's almost like a temporary knockdown experiment. And they then look at what genes are changing in their expression, but from the proteomics standpoint, so using mass spec. So what I'm going to quickly do, because I know we've shown these before, I'm going to run a search using this Plasmodium Bergeye experiment. And as you can see in this experiment, they did 2 things. They compared wild type parasites grown in mice having a normal diet versus a caloric restricted diet. And then also they had this delta kin mutants which kin ends up being a gene that seems to be a central regulator, and so then they compare these also in the different environments. So for the question I'm going to ask, I'm just going to go to the genes based on fold change. And again, I'm going to only compare the wild type. You can obviously do this in different ways, but I'm going to, the question I'm asking is, I wonder if there are any genes that are in caloric restricted conditions in the wild type compared to the control that are, let's just say, upregulated. And let's do this by fourfold. The answer may be 0. I don't think I ran this particular question exactly as it is, but I'm basically asking which genes are upregulated in the parasite when the host is not eating a lot of food, basically. Or maybe that's the normal conditions where the host is in. And so we run this experiment, and see what the results are. So there are 30 genes that met this criteria. And so then I can go down and take a look at them in all the different ways that Suzanne mentioned. So I won't go into that again, but you can look at the graphs here in this table. You can add additional columns to this table and play around with it like an Excel spreadsheet. So the next thing I'm going to do is, rather than going back to the data, I'm going to add a step. And I'm going to look for the quantitative proteomics experiment. So I'm going to select quantitative. I'm going to pick this quantitative mass spec. Here's this new data set that looked at this PFCRK4 knockout line. And it's a direct comparison experiment. Basically, it's a comparison between, plus shield or minus shield. Plus shield minus in different time points. And so I'm going to compare the 37 hours post-infection plus shield to the 29 hours post-infection plus shield. So that's in the absence of kin. So that's one way of doing it. The other way, you can say, well, I want to compare the wild type conditions minus shield to the mutant conditions. This is plus shield. Let's pick that. It doesn't matter. And I'm looking for anything that's differentially regulated. I'm going to run this, and I know the answer is going to be 0. So I'm basically asking which genes are upregulated in conditions of caloric restriction that are also are differentially regulated in this CRK shield conditions. The answer is 0, but the reason here is because, remember, this experiment was done in Plasmodium bergei, and the second experiment was done in Plasmodium falciparum. So I'm really comparing falciparum genes to Bergeye genes, and basically the answer is 0, as would be expected. So you have to have a way to convert one of these into the sets of the other genes, and that's what Suzanne showed us using this orthology tool. And so you can, for example, if I'm interested in working on Plasmodium bergei, I can convert these falciparum ones into Plasmodium bergei. So let's go ahead and do that. I'm going to edit this step. I'm going to click on orthologs up here. I'm going to ask for bergei. So I'm basically saying, for these P. falciparum, the 600 genes in P. falciparum, convert them to the orthologs in Plasmodium bergei. And now, at least we will have the same genes, the same gene IDs, basically. So these are Bergeye now. So these falciparum ones became Bergeye genes. So these are presumably the Bergeye orthologs of the Plasmodium falciparum genes that are upregulated in parasites that were infecting mice that are under caloric restriction. Turns out 9 of those are also differentially regulated in these. 9 of them are also differentially regulated in the parasites that are in the presence or minus shield. And then you can take a look at them and explore them further. Okay, so again, I think this power of orthology is hopefully very evident for you, and hopefully you'll find it useful. The last data set I will describe today is in Plasmodium, which includes data sets for Babesia and Theileria. And we loaded recently Transcriptome and ChIP-seq data set from Jonathan Weissman's group in Paris. And so this is from the same study, but they're loaded as 2 separate experiments because they're different data entities. One is RNA sequence, as we saw before, and the second one is ChIP-seq. So it's histone epigenomic data. And the way to explore this data is we can start with a transcriptome search, for example, just so we can get a list of genes that are highly expressed under these conditions. This was a single condition, so there wasn't any comparison. And so the only search that's available to you is this percentile search. And so I can go in here and say for the transcriptome from infected B cells with Theileria annulata, I would like, and I changed this right before, but I would like anything that's expressed between the 95th and 100th percentile of expression. So if you imagine ranking all the genes in the genome based on their expression levels, I'm saying I want the ones that are above 95 in the expression level. So things that are highly expressed. Okay, so you run this and you get 190 genes that are highly expressed during a B cell infection. That alone is quite interesting, obviously, because now you can potentially find genes that are involved maybe in host modulation or host transformation, and so that's quite interesting, and you can take a look at those. But of course, you may be interested in looking at the histone landscape around these genes, and for that, where we have that is in the Genome Browser, and so I'm going to go to the gene page. That's one way of getting to the Genome Browser. There are many different ways you can get to the Genome Browser, but since I really want to look at this gene in the Genome Browser and explore its histone landscape, I'm going to go to the gene page, and now I can go and view in the Genome Browser. And so this is going to load a default view, but with this gene of interest highlighted in the center of the Genome Browser. So the Genome Browser is very useful for allowing you to visually look at gene models within the genome, and then you can move around, zoom in, zoom out, and I'll show you some of those. I'm going to just go ahead and close these default tracks. I don't care about them right now. So I'm looking at all the gene models here. There are some genes that are in red. Those are genes that are going from right to left in terms of where the gene is encoded on which strand, and then some genes are going from left to right. And you can see there's a little arrow, and those are all the blue genes. Okay. So now I'm going to open up this this window here, select tracks, and I'm going to look for first I'm going to look for the transcriptomic experiment from this, the transcriptomic data from this experiment. So I'm going to click on transcriptomics on the left here, and you'll see here that these are all the B cell tracks that you can load, the B cell transcriptome. You'll notice here that some of these are uniquely aligned reads, some are non-uniquely aligned reads. So what we do is we map all the reads from the transcriptome to the genome, and then some reads will map to more than one place because of gene duplication or stuff like that, and we map them as separate non-unique reads. And this allows you to look at regions that are potentially regions of copy number variation and so forth. So I'm going to pick the unique ones for my purpose, and this is a strand-specific data set, which means that they sequence both strands and they know which one, where the transcripts come from, which is quite useful. We'll look at this in a second. And the next thing I'm going to do is I'm going to look at the epigenomic data. There are many tracks. Again, if you look at the experiment, you can understand a bit more about what's in there, but you'll see that there are a bunch of unique again and non-unique, but also there's input samples, and so I'm going to just load a bunch of these just so we can look at some of that. I'm not going to care about the input for now, so I'll pick a few more of these. And as you can see, as I'm doing that in the background, all of these tracks are loading. I think I'll stop here, so that's good enough. And let's open this up. And so now we're looking at this gene right here, and I'm going to pin this gene to the top so it stays up there. And now you can see there's this gene. It's going from right to left, so it's a red gene. You can see the expression on the reverse strand is high. There's some expression from the forward strand, but it's super low. And then you can look at which histone marks are peaking basically around this gene. And so I can, for example, zoom in a little bit more, and then we can look at this in a bit more detail. So here's this gene, exon 1, exon 2, and you can see there's a peak of this histone 3, the acetylated glycine, basically, in histone 3. And then you can obviously ask the question, well, is this something that's happening in other genes, neighboring genes? So we can zoom out a little bit, and you can start scrolling down and looking, and you'll see here that seems like there's always what looks like almost a double peak, one at the tail end of the upstream gene, and then one at the front of the gene that's being transcribed. Same thing here, right? So you can start finding patterns of histone modification or histone binding. And that's obviously what they were looking at in the paper, but this allows you then to investigate your own data. Okay, so we are at the end of our webinar time, and I think we covered everything that we needed to cover. So thank you everybody for attending. As mentioned before, this has been recorded. I don't see any questions in the chat, which is fine. If you think of something afterwards and you have any questions, definitely drop us a line and let us know about your questions. And hopefully you found this useful, and we will talk to you next time. Thanks, everybody. Bye-bye.
Bye-bye.
Bye.
Bye, everybody.